Welcome, everybody. It's a pleasure to see that so many people have found time to gather here in Egge for this exciting lecture. This is a lecture in the series called Horizonter, or Horizons, I suppose, in English. And these lectures, or this series of lectures, tries to focus on some of the really big issues in current research. And it varies across different disciplines, primarily within the natural sciences, but I can see that it attracts also a lot of attention from outside the faculty of natural sciences and mathematics. That's great. My name is Bergen, and I used to be the dean at the faculty, but now I'm rector at the university, so it's even a greater pleasure for me to welcome you all here to this most interesting talk. It'll be thoroughly presented in a moment, but I'd like to say that Horizons is about the really big issues in science, and genetics is definitely one of those. Since Mendel, this particular piece of science has evolved a lot, of course, and perhaps the revolution started with when the structure of the DNA molecule was described, Watson Crick and Rosalind Franklin, who contributed to it as well without receiving the Nobel Prize. The two guys, of course, received the Nobel Prize for their discoveries. Since then, a lot has changed, a lot has happened, and the Human Genome Project was particularly important, I think. And what's so nice about this project is that it is no longer a sole, let's say, molecular biology research topic. It involves a lot of different other fields, informatics and mathematics as well. I think that is perhaps reflects some of the hallmarks of big science these days, and that is that it takes a number of different disciplines to approach or address the really big issues in science. So again, welcome to this lecture. I'm sure it'll be wonderful.
Good afternoon, everyone, and it is on, I think. I just have to have it close. Good afternoon, everyone, and I would like to thank Rector for opening this event this autumn. Very important for us, and I would like to thank the Horizon Committee for giving me the opportunity to introduce today's speaker. Not just because it is close to my own research interest, but first and foremost because Leonie Ringrose is actually a former colleague of mine and a very good friend, and it's always great to see you here in Bergen again, as it always is. Scientists have known for quite some time that cells in living organisms have a remarkable ability somehow to remember which genes to have on or off. In the beginning, most of the knowledge about this came from studies of genetics, classical genetics, using, for instance, fruit flies, Drosophila. More recently, several other approaches have also been used to gather knowledge about this, and I'm sure Leonie will give us a good idea of what the current state of the knowledge in this very important and fascinating phenomenon is. There are quite a few scientists who could have come here and spoken about this. Leonie is one of the few in the field who have taken a quantitative approach, which is more common among physics and chemistry disciplines and so on. And this is becoming clear to us molecular biologists. That's becoming more and more important because it allows us to build proper models for how the cells are working and so on. So that's going to be rather important in the years to come. I don't know if this is true or not, but maybe this has to do with Leonie's experience when she did her PhD at the IMBA in Heidelberg in the early 90s, where she studied the phenomenon of looping of DNA inside the cell nucleus, which she did with experiments, very advanced genetic engineering, but she also used mathematical modeling to figure out what the data meant. After that, she was exposed to epigenetics already there, but then she went to do a postdoc in Montpellier with Jean-Marie Durand, and after that continued this work back in Heidelberg with Renato Parro's lab at the Centrum for Molecular Biology in Heidelberg, ZBH, using exactly the fruit fly as the key model organism. And since 2006, she has been group leader at the Institute for Molecular Biotechnology in Vienna, where she is still studying the mechanisms of epigenetic regulation using both flies and other beasts. So before I allow you on the stage, Leonie, I'd like to make you all aware that this lecture will be videotaped. There's the camera up there. So you know what that means. And this is done with Leonie's request, in fact. I very appreciate that a lot. I also like to thank the team who do the video, because this was done on a very short notice. This is Anders and Nikolai at the IT department, and also Anders from the Bergen media company. Thank you so much. Leonie.
So the first question is, can everybody hear me? Is the microphone working? Good, okay. So thank you very much, Rain, for that very nice introduction, and also for the introduction to the entire lecture series. So I really want to start now by thanking Rain himself again, because a year ago he suggested my name to take part in this lecture series. And I want to thank Dag Aksnes, who's the person who actually invited me and organized all of this. And it's a great honor and a real pleasure to be here this evening and to share some of my thoughts with you about epigenetics. But before I do that, I want to tell you a story. This is the story of Princess Isabella and Prince Harry. Every day, Prince Harry would go to Princess Isabella and tell her how much he loved her and why. He said, "I've heard that you have magic powers. I've heard that you can sing the blues. I've heard that you can cure diseases just by snapping your fingers. And I've heard that you can even make beautiful babies out of ugly parents." One day, Isabella had had enough. She got very angry. She stamped her foot and she said, "As many people hate me for those reasons as people who love me. And anyway, none of it is even true. I challenge you to go out and find the truth about me. And don't come back until you have found out 10 things about me which are both true and interesting." And so, our hero set off on a journey to find out the truth about Princess Isabella. He traveled far and wide, and he asked many people. He traveled, and he did his research for a year and a day, until finally, he came back to Princess Isabella and asked her whether he had passed the test. He said, "I've found out that you like lemon ice cream. I found out that you can ride a motorbike. I found out that you're an expert in juggling. You like reading Mickey Mouse comic books, and I found out that you love playing poker. And most important of all, I've found out how interesting you really are." Now, Princess Isabella was very happy that Prince Harry had finally understood her. And ever since then, they've been the best of friends. And Prince Harry knows that Princess Isabella is the most interesting person he's ever met. And every new thing he learns about her just tells him how fascinating she really is. And that is the story of Princess Isabella and Prince Harry. And now I'm going to tell you another story. This is the story of epigenetics. And I'm going to show you this evening how I think the story of epigenetics is really very similar to the story of Prince Harry and Princess Isabella. What do we mean when we talk about epigenetics? Now, the word epi in the word epigenetics comes from the Greek, which means over, above, or outer. So we're talking about something which is on top of genetics. Now, if you look in Wikipedia, you will get this definition. Epigenetics is the study of changes in gene expression or cellular phenotype caused by mechanisms other than changes in the underlying DNA sequence. And I want to illustrate this with an example that you might all be familiar with. The caterpillar and the butterfly share exactly the same DNA sequence, but they look very different. And this is what we mean by a change in cellular phenotype. Now, we also know that to make that change happen, there are lots of genes being switched on and off. So that fulfills the definition of a change in cellular phenotype, which also involves changes in gene expression. But those two forms of the animal have completely identical DNA sequence. So this is a change in gene expression and cellular phenotype without a change in the underlying DNA sequence. But what makes it epigenetics is this word, the mechanism. And that's what we're going to talk about this evening. And to introduce you to that, I want to introduce you to the epigenome. Now, just as epigenetics is talking about something on top of genetics, the epigenome is talking about something on top of the genome. And so the genome, when we talk about this, what we mean is all of your DNA. And that includes all of your genes, and so all of your genetic material. And that's what we mean by the genome. Now, the epigenome is really made of molecules that sit on top of the DNA. Some of them are more closely attached than others. And during this lecture this evening, you're actually going to meet some of those molecules, and you're going to get to know them quite well. So I will tell you about that later. But right now, what I want to do is just to talk about what is the effect of the epigenome on the genome. Now, the DNA is nothing more than a text. Now, in the language of DNA, we only have 4 letters. So it's a very special language. But you can imagine it as a text in any language you choose. And so you can imagine it as a text in English. The thing that this text and the DNA text have in common is that they both have information. And now you can imagine the epigenome as something that formats that text and makes some things more important than others. And so here, for example, if you imagine we're in the palace of Princess Isabella, she might think that certain things in that text are more important than others. So here's how she would format that text. But if we're with Prince Harry, he might do it differently. And so you can imagine the epigenome as something that formats the same text to give different interpretations in different situations. And if we then go back to biology, that means, for example, that the caterpillar and the butterfly use their epigenome to read the genome in different ways. Or you might imagine for yourself that your brain cell and your liver cell have different formatting on the text of your genome. Okay? Now, there's another thing that the epigenome can do, which makes it very interesting, that Rain also referred to. And that is, it can remember how it formatted that text, and it can do the same thing over and over again. Now, in your body, actually, let's ask a question. Does anybody know or want to guess how many different cell types you have in your body? Any guesses? Do you think it's 50? 300? It is. It's between 200 and 300. It's a lot. They're different. And many of those cells are dividing all the time. They're dividing and dividing, meaning that they make new cells whenever you need them. Now, we know that the DNA inside each of our cells is identical in the daughter cells. It's exactly the same thing. But it turns out that the epigenome can also be passed on to the daughter cells. Now, why is that useful to you? Why should you care that your epigenome is being copied? Well, I'll tell you. Let's think about your liver. Now, during the time that you were a little baby and you were growing into an adult, and even right now, your liver is making new cells. And those new cells have to be liver cells and not brain cells or any other kind of cell. And the epigenome can help your liver to do this, and all your other cell types as well. And this is the concept that we have in epigenetics, which we call epigenetic memory of cell identities. And that helps to keep your cells different from each other and correct. But there's another really interesting thing that the epigenome can do. You have a liver for a reason. It's good for something. And as many of you probably know, one thing that your liver is really great for is for dealing with alcohol. Now, if you drink a beer or two, what will happen in your liver is that certain of your genes in your liver cells are going to react to that so that they can take care of the alcohol that's coming into your liver. Now, what you may not notice is that while it's doing that, it's going to change its epigenome for those particular genes. So the epigenetic marks will change. And now, because we know that the epigenome can be copied, it is entirely possible that the new cells in your liver will carry those changes even after you have cleared the alcohol from your system. And then maybe the next time you have a beer or two, you can deal with it even better. I'm sure many of you have noticed that effect. It's a training effect, isn't it? So, this is a hypothetical example to give you the idea of how it might matter to you, but there are examples of specific genes where there is an epigenetic memory of something that happens during your lifetime. And we call this epigenetic memory of life experience, if you like. Okay? So there's two different things. There's the cell identity that stays the same. Your liver cell doesn't change into a brain cell when you drink alcohol, unfortunately. It stays a liver cell, but some of its genes change because it can cope with the alcohol. Okay? So epigenetic memory, two sorts. Now, we've been talking about a kind of inheritance. What we've been talking about is information that gets passed from one cell to its daughter cells. But does it go further? Can we actually imagine passing epigenetic information from parents to their children? Now, we know that children look like their parents because they get their genes from their parents. And it's probably clear to everybody in this room that however hard I try, much as I might like to, I will never give birth to a family of badgers like this, because I simply don't have the right genes. But if we're interested in epigenetic inheritance, then the question that we need to ask is, if I or this badger drink a lot of alcohol during our lifetime, and that makes changes in our epigenome, as I've shown you before for the liver, will that then have an effect and be passed on as epigenetic information to our children? Now let's just think about this. If I make epigenetic changes in my liver, that is completely irrelevant to my children, because we don't make babies from our liver cells, okay? We make babies from sperm and egg cells. Everybody does it that way, except maybe yeast or whatever. Anyway, mammals make babies from sperm and egg cells. So what we need to ask, if we're interested in epigenetic inheritance of life experiences, is, does the thing that we do during our life make an epigenetic change in our sperm or egg cells that is passed on to the children? And remember, the babies are made from one cell. So every single cell in the body of your children will have the same epigenetic information. So the question is that then relevant for the thing that we're interested in? For example, the function of the liver. So that's just to set up the question that we should be asking if we're interested in this. Okay? Now, this is obviously a very interesting question. It's something we'd like to know the answer to. And there are a lot of people out there in research working on this. And I'm going to introduce you to two of them. These are two scientists who are working on epigenetic transgenerational inheritance. And here is what they say. Emma Whitelaw says that this phenomenon, the transgenerational inheritance via sperm and egg, is likely to be rare. And she says it's likely to be rare because of this fact that I mentioned, that any epigenetic change in the sperm and egg is going to be present in every single cell of the body of your children. And that can cause an enormous number of problems. So it's likely to cause problems for the animal. Okay? Oliver Rando says that there are examples, and he talks about this, there are examples where you can have transgenerational inheritance of environmental influences, but the mechanism, how does that actually work in molecular terms, is not clear. So we don't know if it works by truly epigenetic mechanisms. Now, given this uncertainty, it's quite surprising that just the idea that this might be theoretically possible has caught the imagination of the world's press. In 2010, this article appeared in Time magazine, talking about why your DNA is not your destiny. And since then, many similar articles have appeared, which seem to totally accept these claims as the absolute truth. And the main claim of this article is here. The author says that the new science of epigenetics reveals how the choices you make can change your genes and those of your kids. And there are a lot of articles that believe this. Now, one of them, this reminds us of Prince Harry and why he was so in love with Princess Isabella, because she was so fabulous. Now, the article says that epigenetics could help explain certain scientific mysteries that traditional genetics never could. Now, I would like to take some time during the discussion after this lecture, and I'll be very happy to go through more examples from this article. I just want to jump now to the main message that the author gives at the end of the article and give you some idea of where the article is going. The author concludes, the potential is staggering. For decades, we thought DNA was an ironclad code that we and our children and their children had to live by. Now we can imagine a world in which we can tinker with DNA, bend it to our will. Now, Princess Isabella would disagree with that very strongly, and I would agree with her. And many people in the field would also be rather disturbed by these kind of claims. And I just want to introduce a few of them to you now. Adrian Bird is an expert in epigenetics who's been around for a long time, and just last week he published an article where he questioned these claims. He says, why should acquired obesity, diabetes, or impaired learning and memory be fostered on the next generation? Most of these epigenetic changes are bad. Why would we give them to our children? He also makes the very important point that in biology, ideas are relatively cheap. But it takes rigorous testing and time and ingenuity to test them. And this idea is lovely, it's out there, but there really is hardly any evidence. Now, Edith Hurd is another person who feels very strongly about this. She's also a scientist working on epigenetics. And she recently gave an interview in the British newspaper, The Guardian, where she puts the record straight. And what Edith says is, even our epigenetic changes are genetically driven. There is only one code, and that is the code of DNA. But now with epigenetics, we're hoping we can pray our way out of faulty genes. Now, she also mentions the fact that epigenetics is exciting. But there's a gap between the fact and the fantasy. It's been overhyped. And I would agree with Edith on that point. And Princess Isabella would agree with both of us. Now I think, if we're going to really do justice to epigenetics, we're going to have to take up Princess Isabella's challenge and go out and find the truth. And that's what I'm going to do with you this evening. I'm going to take you on a journey. And like Prince Harry, I'm going to take you to find out some of the truly interesting things about epigenetics. Now, the first place I want to take you is the cell nucleus. A typical cell in your body will have a cell body like this and a small nucleus in the middle, and that is where all of the DNA is stored. Now, if you took all of the DNA inside a nucleus of one of your cells and you stretched it out like this, does anybody want to take a guess or have an idea how long that would be? Any ideas? 2 kilometers? Any advance on 2 kilometers? Over here. To Pluto? We're talking about one cell here, not your whole body.
3 times around the world? 3 times around the world?
3 times around the world. No, it's actually only, oh, go on. 2 meters, he's right. The man at the back is right. It's 2 meters. We're talking about one cell there. So those of you who were thinking about Pluto, yes, if you took all of the cells in your body, definitely. But for one cell, it's 2 meters. Now that might not seem like very much, but I'll give you another illustration. It's about the same as if you took a piece of string 20 kilometers long and you tried to pack it inside this juggling ball. So that's quite a big task. And this evening, you're going to experience actually how that works. And for that, you're going to need some training. So I would like you all to stand up, please. Stand up. Okay, now I want you to turn to the person next to you on either side and shake their hand. Shake their hand. Okay, on the other side, shake the hand. Okay, very good. Now turn to the person on your left and pat their shoulder. We're going to be collaborating this evening, so you need to, yes, very good, very good. Okay, very good. Now, we're going to try some voice exercises. I am not going to ask you to speak, but this is just to focus you on the task in hand. When I count down from 3, 3, 2, 1, I want you to shout DNA. So, 3, 2, 1, DNA! Very good. Okay, let's do epigenome. 3, 2, 1, epigenome! Very good. Okay, super. Now, you can all sit down. Now I need one volunteer from the audience. This won't be the last time I ask for a volunteer, but you will be the first volunteer. Do I have a volunteer? Oh, thank you. Come to the front. Wonderful. Come here. What is your name?
Sindre.
Sindre.
Yeah, Sindre in Norwegian.
Sindre is going to be he's going to be the DNA man this evening. So can we have a big hand for Sindre, please? Thank you. So, what I want you to do, Sindre, is take the end of this very long piece of DNA. It's not 20 kilometers, but can you walk with it as far to the back of the auditorium as you can? Just walk up there. Thank you. Thank you very much. Up he goes. So you can see how long this is, okay? Right, stop there, don't leave the room. Okay. Now, what we're going to do this evening is get this entire molecule of DNA packed onto the stage down here in front of you, but in the most organized way possible, because we need to always know where every part of it is. Now, does anybody have an idea how we might do that? Not the people who know how it's done, but any idea? How could we package this up? Any thoughts?
Buckets.
Hmm?
In buckets.
In buckets, yeah, you could put it into little containers. What else could we do? We could hang it up. Anything else? Well, I hope if I ask you this question this evening after my lecture, everybody will know the answer, because we're going to do this right now. Okay, and for that, I need another 10 volunteers. Yes, come down the front. 1, 2, from 3, 4, great. 5, please come down. 6. Yes, just come down until we've got 10 of you on stage. If we can fit more, we'll fit more. Can you line up in the front here, please? Wonderful, thank you. Come and line up. Make a nice line up at the front. Okay. All right, how many have we got? We got 10? Yeah? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Anybody else? There's a little bit more space. No? Okay, we'll go with 10. Now, I'm going to show you. You have to watch me. I'll show you what I want you to do. Okay? This is safe to try at home. You can do this with your friends, as long as they're good friends. I want you to take the piece of DNA and wrap it exactly twice round your body. So, let's see. 1 and round. Yeah, this is not obvious. Twice round. That's once, okay? And then twice, like this, okay? Twice round the middle, and then onto your neighbor. So, what I want you to end up with is something that looks like this. This was the part where I wasn't sure how long it would take. If you get stuck, look behind you. There's a diagram of how this should look. Each one of you is one of these molecules, okay? You don't need 4 inside one. Yeah, very good. How are we doing? We getting there? I told you you'd have to collaborate this evening. Wonderful. Can you move? It's looking good, huh? Move further along, yeah? Move along. Yeah? How are we doing? Okay, very good. Now, these people at this end, while they're finishing up over there, can you move a little bit over and move a bit further apart from each other? Let go of the rope and let it go out. Let this out a bit. Yeah, very good, very good. Give yourself more space. Very good, very good. Very excellent, yes. How does it feel? Yes, okay. Okay, ladies and gentlemen, we've just created human chromatin. Now, this is exactly, almost, how the DNA is packaged inside the nucleus. This is how we get the DNA into a smaller space, but still in a way that we can access it, and we know what's there. But this is the same across the entire genome. So now, I'm going to give you a live demonstration of the epigenome. So where's my DNA man? Can you come down and help me, please? Bring the end of the DNA with you, or leave it there, doesn't matter. Okay. What I want you to do is, starting at that end, can you give out these cards? Give each person one card. Okay? Now, hold on to your rope with one hand and hold up your card with the other hand. Up in the air. Perfect. Give him a red one. Okay. You are the epigenome. This is the epigenome, but it's not yet doing anything. So what I want you to do now is, if you have a red card, shuffle up nice and close to the person next to you. Get nice and close. You can hold each other's ropes if you want to. Don't let go. Hold on tight. Get close. You can even squeeze in more tightly. Get into a bunch. You can fold around. Yes? Wonderful. Yes, yeah, perfect. Yeah, wonderful. Okay, there we are. If you have a green card, I want you to hold on to your rope with one hand, but step out of it. Get free. Wonderful. Hold on to the rope with one hand, though. Stay there. Hold on. Yes? Perfect. Okay. Now, you can see that the genes in this area where the green cards are going to be very easy to read. They're going to be accessible. And so it's very easy to switch them on. But the genes over here in the red part, nobody's going to be able to read those. Those genes will be silent. So that, ladies and gentlemen, is exactly how the epigenome works inside your cells. Now, there's one more thing I would like you to do before we send these people back to their seats, thank you very much, is let's give out the prizes. Okay, thank you very much. Now, you've just experienced how it is to be inside the epigenome. And I just want to introduce you to a few words which we're going to be using in the rest of the talk. The people wrapped up around inside the rope are called histones. It's a kind of protein. The people who, together with the DNA, the people and the rope is called chromatin. And I'm going to be using those words in the rest of the presentation this evening. Now, what you've just done here on stage is something that scientists do a lot. You've just built a working model of the process you're interested in. Now, a good model will help you to understand how something works. And a really good model will help you find out which are the questions you should be asking. And I must admit there were some new questions that I hadn't really thought of at all that arose while we did this evening. But there are two questions that come up from that model you've just made, which are really some of the most important questions in epigenetics. And that is, who gets those cards and who gives them out? Now, if we think about this first question, who gets the cards? As we sit here right now, there are billions and billions of dollars and euros and other currencies being spent on the effort to map where those red and green cards go in the genome of different animals and different cell types. There is a vast amount of data available to tell us where they are. And the interesting thing is that they are not all over the genome. Those marks, I call them marks or cards, if you like, are very, very selective about where they go. Now, what these cards or marks are really made of is chemical groups which get attached either to the DNA or to the histones. And they go only to certain places. And where they go is different in each cell type, but where they can go is determined by the sequence of the DNA. Now, you can think of this like traffic lights. We don't have traffic lights all the way along the road. We just have them at important junctions, and that controls what happens in between. Okay? Now, another question where there's a very large amount of active research is who gives these cards out? And there, we've made a lot of progress in the field in identifying specialized families of green and red proteins. Now, the green proteins are the ones that give out the green cards. They're what we call activators, and you've seen how that leads to gene activation. And the red proteins give out the red cards. Okay? Now, these proteins are enzymes. That means they catalyze a chemical reaction, and that's how they join those small molecules onto the histones or the DNA. Now, you can just imagine an enzyme, or these proteins, as a big molecule that's going to place a small molecule onto another big molecule. So, these are enzymes, and there is a vast amount of research being done there. And we know quite a lot about different enzymes, how they work. Now, also, the whole process is reversible. So, there are other enzymes that will take those marks off, so the whole thing can change whenever we need it to. Okay? Now, those are things we know a lot about. We know what the epigenome is made of, and we know how it gets onto the DNA or the chromatin. But there's a very big mystery that we have not been able to solve. Now, I told you that the epigenome gets copied, and the question is, how does the epigenome get copied when the DNA gets copied? Now, we've known for a very long time that DNA can replicate or copy itself because it can unzip its 2 strands. And each one of those 2 strands, which are shown white here, those are the parental strands, each one can assemble and be used as a template to make a new strand, which is shown in black here. And the beautiful thing about this whole process is that these 2 double-stranded molecules are now identical copies of the parent DNA, and those are what get passed to the 2 daughter cells. Now that arises from the structure of DNA. But I've told you that the epigenome can also get copied. And it is absolutely unclear to us how that happens structurally. What is the molecular process? It's not obvious, but it does happen. So for the last part of the talk this evening, I'm going to take you on another journey. And I'm going to take you now through some of the research that we've been doing in my own group to try to address this problem of how the epigenome gets passed on to the daughter cells. Okay? And the first place I'm going to take you is deep inside the brain of a Drosophila fruit fly larva. Now, you will be very relieved to hear that I'm not going to ask for volunteers for this. Instead, I'm going to take you to the movies. Now, the first story you're going to hear this evening is a story with 4 heroes. The first hero of this story is the green fluorescent protein. Now, this molecule won the Nobel Prize in 2008. Well, actually, the people who discovered the molecule won the Nobel Prize. Why is that a hero? Now, that protein is a hero because it's a protein. That means we can attach it to other proteins inside a living cell. And because it's green and fluorescent, we can actually see it with a special microscope. And that means we can label proteins inside a living animal and watch them doing stuff. Now, the second hero of the story is our fruit fly, Drosophila melanogaster. Now, the epigenome of Drosophila melanogaster works in exactly the same way as our epigenome. But it's much easier for us to fix up the proteins of this animal with this green fluorescent protein and label them. We cannot really do that in humans, or we can't do it without breaking laws. So we can use the fruit fly, if you like, as a model for the processes we're interested in. Now, the other 2 heroes of this story are 2 wonderful people that I had the great luck to work with, which is João Fonseca and Philip Steffen. And they are the guys who worked out how to use this protein in this animal to try to answer this question. And that's what I'm going to show you now this evening. Now, I'm going to show you a movie of a real Drosophila cell actually dividing. You're going to catch it in the act. This is a real-life movie. And what you're going to see here are 2 things. You're going to see a green protein, which is one of those activators. And this green protein is labeled with green fluorescent protein, so it's green in the movie. And here it is. Here is the cell we're interested in. You can already see that protein there, okay? But the other thing I'm going to show you, and that you have to watch out for, is some red stuff. And what we've done is we've used a different version of that green fluorescent protein, which is red. And we've labeled the histones themselves. So you're going to actually see the chromatin. So while you watch the movie, you have to watch out for the red and the green. And what we want to know is what happens to those green proteins when the cell actually divides. And you're going to see it go through a cell division and make 2 daughter cells. So watch out for the green stuff. But we also want to know what happens to our chromatin as the cell divides and goes through and makes the 2 daughter cells. So watch out for the red stuff. And I can play it more than once. So let's do it first time. Okay, you see at the beginning something's moving. The green stuff. So now the red stuff becomes apparent, the cell divides, and the green stuff disappeared but comes back. Let's show it again. The green stuff's there. Now the red stuff appears, the green stuff disappears, cell divides, green stuff comes back. Okay? So what you need to remember from this, for the next part, is that when the red stuff appears, it's not just coming from nowhere. What's happening is it's squeezing up tight. It's packing up. It was very dispersed, and now it's becoming very concentrated. That's why you can see it. It's squeezing into a small volume. So let's just show that one more time. And watch only the red stuff now. There. It's very small. It gets bigger again, okay? And you're gonna now see that in the next movie that I show you. So this was a movie of a real cell. So this is a real image. And what we did then was we did this with many different cell types, and we labeled many different proteins, and we put this then all together into a model, which I'm going to show you as another movie, but be aware that this is now an interpretation. And to get from these movies to the representative model, we used a lot of mathematical modeling in between, and that's all I'm going to say about the mathematical aspects. I'll be very happy to talk more about that, okay? So, the next thing I want to show you is an animation showing some highlights of what we found out from that process of filming these cells in action. Now, what you're going to see here is these green green activating proteins and red repressing proteins. And you want to see the chromatin. Now, at this point, I should point out that I've changed the colors from the previous movie, so forget the red and green from that movie. What we've got is, in the movie, the activators are colored green, the repressors are colored red, and the chromatin is colored blue like this, okay? Now, what you will not see in the movie, here, in this animation, is those cards, those marks that these proteins make on the histones. And I've just left them out to keep it simple, but you can imagine that whenever these proteins are near to the chromatin, they will be making those marks, and they will be either activating or repressing their nearby genes. Okay? Now, this is the last movie I want to show you, so just sit back and enjoy it. Now, the first surprising thing we found when we did this analysis was that these proteins are actually amazingly dynamic in the way that they interact with chromatin. They're hopping on and off their target sites, and they never stay for long. Now, if you look closely, you'll see that even the red and the green proteins are competing for the same pieces of DNA. So how can this give any kind of stable regulation? Well, it turns out that one thing that's important is how long do those proteins stay when they sit on DNA? If you're in a situation where the red protein stays longer, it will make more red marks on the histones, and the genes nearby will be repressed, even though the green protein visits. And if the green protein stays for longer, it will make more green marks on the nearby histones. The genes will be activated, even though there is an occasional visit from a red protein. And this very dynamic but regulated series of movements is important for the way that the epigenome is regulated. Now, this could keep the epigenome happy forever. But every few hours, there is a major challenge to this situation, and that is cell division. So during cell division, as you've seen in the previous movie, the chromatin is going to condense and squeeze together, and many proteins will actually come off. Now, what does this mean for our red and green proteins? Well, it turns out that the red protein stays on the chromatin and actually almost completely stops moving for the whole time of cell division. But the green protein binds, but hops back and forth. It keeps hopping. So both the red and the green proteins are bound to chromatin all the way through the cell division, but they bind in very different ways. Now, what does this mean for the cell in the next generation? Does that help the cell to remember what was happening before? Now, we think it does. And what we think is that the sites where the red proteins were bound very strongly during the cell division, those are the sites where the red protein stays longer in the next generation, wins the game, and silences the genes, even though the green ones are trying to get in there. And the places where the green proteins were are the places where in the next cell generation they will bind longer and they will win the game against those red proteins who are trying to repress. So it's a very dynamic situation. And we think that this dynamic system can survive even the huge disruptions of cell division. And if we can understand more about the molecular details of this, then we may be closer to understanding the key to the molecular mechanisms of epigenetic memory. So, we have come to the end of our journey this evening. And I hope that, like Prince Harry, you've found out some things about epigenetics which you might find interesting. And I hope that, like me, you now may have the feeling that the really, really interesting things about epigenetics are those molecular details which are not immediately obvious, but which are what really make the whole field fascinating and exciting. So it just remains for me to thank a few people. I want to particularly thank Philip Steffen and João Fonseca. Those were the two PhD students who've now finished their PhDs and moved on, who did the work where I showed you what we've been looking at inside the fly cells. I want to thank Vari Taula, who's the director of the company that made the animation, and she stayed up until midnight last night to send me the final version, so it's really hot off the press. You're the first people who have ever seen this. I want to particularly thank Mark Reemsmeijer, not just for providing the climbing rope and the juggling ball, but also for constant inspiration and many discussions and many other things. And I want to show you these people. So in science, we never work alone. We need to have other people in a group working with us. And these are the people who are in my group at the moment. And they're the people who really keep it going. They ask the questions, they do the experiments, they do the work. And they keep me great company all the time. So I want to thank those people. Finally, science is not free, and I want to thank all of these organizations for providing money for this. And thanks again to the organizing committee for inviting me. And while I take the questions and we run the discussion, I want to leave up this slide, because this is a source of some really interesting writings about this subject. This is a book which is written by a scientist, which is factually correct. So if you want to know a lot about epigenetics, read that book. These are the two articles I cited from Edith Hurd in The Guardian and from Adrian Bird in the journal Cell. And this morning I discovered that there's actually also an article in Norwegian. So if you want to read about this some more, there's a very nice article about not only genes, but as far as I can work out, right, from the Norwegian version, it's also about epigenetics, right? Yes, okay, so this... Interesting and true, yes. Okay, so I'm very happy to have had your attention for this time, and I'll be very glad to take questions.
I knew it. It is possible to be a great scientist and a great storyteller, and she is actually right here. Okay, you're happy to entertain some questions? And please raise your hand. We have a not only walking but running microphone over here. So I'm sure there must be questions. Yes, please, in the back. Oh, good exercise for you, right?
Can you hear me? Yeah. Thank you very much for your fascinating presentation. Yeah, my question is, you indicated in your talk that the two states of genes are either red or green. And my question to you is there a third, or are there other options? Is it either activated or repressed, or is there something in between?
That's a great question. Did you know that question already, or did you get that from watching the model? Great, so that means it was a good model because it came up with another good question. Yes, there are. There are states in between, and there are different shades of red and green. So I simplified it down to active and silenced, but there are also half-active states, which can be a mixture of red and green. There are also red marks which are more strongly silencing, and other red marks which are not as strongly silencing. And there's a very interesting situation in stem cells where we have something that is called bivalent chromatin. And on that chromatin, we have both red and green. And the idea there is that that chromatin is undecided because it's an embryonic stem cell. And when the cell divides and takes up, say, two different fates, it goes towards the brain or the liver, one of those marks will be erased and the other one will stay. And all the cells that you don't need all the genes that you don't need in your brain will be silenced. And the same for the places where you have the green mark, we will take away the red one and keep things active. And that's, the idea there is that the chromatin is poised, it's ready to go, it can become anything. And there's a lot of research going on this idea of bivalent chromatin. Yeah, so your question is a really good question, and it has a very complicated answer. But yes, you're right, there are things in between. Yeah. Mind the rope.
So I'm not sure if it was your saying or if you just quoted, but you said something about the epigenetic changes is only bad stuff. Could it not be good stuff, and that it has that it is something that would be nice to inherit?
Yeah, there is one example of that, as far as I know, which is convincing and a good experimental proof. And that is in the worm, C. elegans. It's a worm that we use for experiments. It's recently been shown that the worm can acquire immunity against diseases, and that it can pass this on epigenetically to the daughters of the worm. And that works via small RNA molecules, which can be considered epigenetic information. In humans, the equivalent of that is known, which is that mothers give immunity to their babies through their milk. Acquired immunity, they can pass antibodies on through their milk to their babies. But that is not an epigenetic mechanism in the way we've been discussing, because it doesn't go through the egg into the animal, it comes through the milk, so we do it. And immunity is something which would be a good thing to pass on epigenetically, yes, definitely. But the front of the field has not yet discovered in humans epigenetic transmission of immunity, but in worms, yes, and plants.
I have a question that is related to the first one.
Where are you?
Here.
Ah, hello.
What is the time frame of this dynamic process of hopping on and off proteins? How fast does it go? Is it milliseconds, minutes?
It's under a second, yeah, it's less than a second. So the difference between the one that stays a long time and the one that moves very fast is a second down to milliseconds. But what I showed you when the cell goes through the cell division and the red protein stays sitting there, then we're talking about several minutes. Okay, so the whole time that the chromatin actually stays condensed and going through this cell division is several minutes, and the red protein doesn't move at all during that time. So that is an enormous difference. Yeah.
Back to the point about the epigenetic changes being harmful. You could take mutations and say most mutations are harmful. therefore we should not see them. But we see them because they're good for the organism as a whole to change to the environment. So why couldn't we say the same to the epigenetic changes?
Yeah, I think we can. I think that maybe I said that too strongly. These are the arguments which are being brought by the people who are writing about this at the moment. Because, yeah, it's a historical thing. Because the examples of transgenerational inheritance of environmental effects, they happen to be mostly negative. That's why. Yeah, but you're absolutely right. Theoretically, there's no reason why we shouldn't have a positive effect and that could be passed on. It's not the fact that it's epigenetic that makes it necessarily bad. It's the fact that there are a very small handful of examples. For example, a period of starvation in the parents can lead to obesity, or it's correlated with obesity in the next generation. And so that's just one example where it's something that you can see and notice. But you are absolutely right, yes. They don't have to be harmful.
You mentioned that, well, we do know how DNA is copied. We know that for quite some time, but we don't know how the epigenetics, how do you actually conduct experiments to sort out these things?
I'm
sure the question
difficult.
will be very,
yeah.
Oh, it's difficult, is it? Okay.
It's extremely difficult, and it's more difficult than some other kinds of experiments. Okay, I should separate the two things that we have to look at. One is we have to look at the copying mechanism, which is, as I showed you with DNA, we have a copying. And then, after that, there's the stage where you share out those two molecules to the daughters, and we call that replication and mitosis. And those are two completely different questions and completely different experimental approaches that we have to take. What I've shown you with our experiments is mitosis. So that's one way we can study, by really taking films, labeling the proteins, watching what they do, but that's descriptive. That says we know where they are. It doesn't say that it matters. Of course it matters to me because I love those molecules. But does it actually matter to the animal? So what you've got to do then is, once you've seen the process happening, is design an experiment where you stop it happening. And another study that I didn't tell you about, we've been able to actually disable those green proteins so that they no longer can bind the chromatin during mitosis, and then we watch what happens to the animal. And the animal is dead, actually, which is a great result, because it means it matters to the animal. Yeah.
For the wet, I think it's a great result with that. Then I'm not going to have another question over there. Yes.
Yeah, I was wondering with the transferring to your offspring, has anyone thought about whether this would happen through females or males, since the eggs are already made when a child is...
Yeah, that has been studied very well. The two people, Oliver Randall and Emma Whitelaw, that I cited to you, they have published stuff on this, so they're probably the two experts. There's a difference in the way you interpret the experiment. If you do the transfer via the female, then of course the mother is with the baby, right? At least until it's born. Even in mice, you still need the baby to be with the mother. So then it's very difficult to distinguish the effects of culture, if you like, from the real effects of epigenetics. So people have then also done experiments with the father in mice as well, where you really take the father away as soon as he's mated. But it turns out, even that, there is a cultural effect, and that's been shown in birds, in finches. If you paint the head of a male finch in different colors, different finches, different colors, you let him mate with the female, and then you take him away. The female that has mated with a finch with a one-colored head will take care of her eggs differently than the one who mated with the male finch who had another colored head. So the female perceives, how important was that male to me? How much care am I going to take of that male's children? So it's really hard to take those things apart, right? But yes, it's a very important issue, and there are studies that have been done on this, yeah.
Thank you. Thanks for a lovely lecture. And I hope I can have a question. You used this analogy of the small chemical things, the marks, the red and the green things. When you showed, let's say, Isabella's text with the highlighting of the text, if I came to see your book, it's me perceiving with my eyes that allows me to see that highlighting.
Yes.
Can you, of course, I have some idea about this, but maybe it's completely wrong. How does the cell actually see the marks that you're talking about? Any idea?
So this is really interesting because I was having breakfast this morning. And I was talking to Mark. And I said, I bet Rain asks the question about how do the marks get read? And actually, on that slide where I had the 2 questions, I originally had 3 questions. And then I realized this is going to take way too long. Yes, so there are readers of the marks. If you imagine yourself standing here as a row of chromatin, and I tell you, if you have a red card moved together, you don't do that on your own. There are other proteins in the cell that recognize those marks, and they will bring you together. And that's another big field of research that I didn't mention at all. Because, I'll tell you why, it's actually in my field of research where I work on a family of red and a family of green proteins, it's the same proteins that put the mark that then actually read it. So it's a self-fulfilling system, and that I felt was too much. So yes, but of course we mustn't forget the readers, the people who read the text. Yeah.
2 very short last questions. Epigenetics, is that good news or bad news for social sciences? And the last one, now we've had genetics, we've got epigenetics. What's beyond? 2 very short questions.
You want short answers to those? Okay, the answer to the first question, I will give you a question back. Do we have any social scientists in the audience? Okay. Even if you're not a social scientist, do you think that epigenetics is good news for social science? Put your hand up. If you're a social scientist. Okay, a handful. Who thinks epigenetics is bad news for social science? No, so the answer is clearly yes. It's good news. Good, okay. And what was the other question?
What's beyond epigenetics?
What's beyond epigenetics?
Okay, if you can't
Any
question,
suggestions?
if you can't answer the question now, we'll invite you back in a couple of years.
Good,
I'm sure
okay.
you have the
And I'll have
results
some
by
ideas.
then.
I might know
Thank
it
you for
tomorrow
coming.
morning.
An umbrella because it
thank you.
rains in Bergen.